So we when we set out with this campaign of 1 million bioinformatician campaign, we never envisaged that we will get this kind of response. So we we were so happy that wow. So that means what we are doing, people saw reasons with it, and people want to associate with this kind of endeavor, and we're like, wow, let's keep on pushing forward, and it's been a very wonderful journey so far.
For sure. It's a very noble goal for humanity.
Yeah.
Um, a quick one, Professor.
Go ahead.
Yeah, I just want to comment. You have a beautiful smile before you start the presentation from your GitHub
Thanks.
profile. Your GitHub profile, actually.
I feel like I look like a deer in the headlights because it was in the middle of a conference that I was moderating, and they basically just pulled me aside and said, "We need a professional picture, stand here." And so when we got the picture back, my husband looked at it and he goes, "Your eyes, you look like you're going like I was doing 4 things when they took this picture."
Yeah, actually, I think I agree with him. And it makes me wonder, a kind of smile. It's hard to have a bioinformatician have that kind of smile.
Hey, what are you trying to say here? Wow,
I'm trying to say we both don't have that kind of smile. I'm still upcoming and you are there. Austin, can you smile? Let me see. See? We both don't have that kind of smile, so I'm just kind of curious. A beautiful smile for a very talented
I actually have something
bioinformatician.
similar to that.
Hey, Ashraf.
Not up to Professor Smile, but I still I dabble a bit. uh, you're welcome, everyone. Sorry, we are about to start. We're waiting.
Thank you for joining.
For the time to clock before we begin officially. So you can feel free. You can see it's just we are having an informal session right now, and you can feel the mood in the atmosphere in the air, and it's as if we are in between a class between our professor and the students. So feel free. You're welcome.
It's Saturday.
And it's almost Sunday here in Korea.
It's the weekend.
It's Sunday in our time. It's the weekend.
Yes, the session will be recorded.
Yes,
I had the same question. I'm just admitting anyone that pops on the screen at this point.
Yeah. Please do. So for those who are here, please inform those of your friends who are yet to be here that once we reach the capacity of the Microsoft Teams, so those who are not here will not be able to join, so they will have to join through the Facebook link, through the live Facebook live video. So please.
We might want to post that link in the chat so people can give it to their friends.
Okay, definitely. I'll do that right now.
Thank you. Here they come.
All right, guys, thank you for joining. Cyan Roy, thank you for joining. So we currently having 3-minute recess. before we start the program proper. So I can see the seriousness is already
Setting in.
coming up. Yeah. Personally, I really don't like it serious. But when my code is not running, then I get serious.
Sometimes the best thing to do is just go for a walk or take a shower.
Yeah. Most time I go to the field and look at the open sky.
There you go.
And most times it usually solve the problem.
Yeah.
Yeah, it does. It does. My kids play Sims, which is kind of this character-based, try to get your character through real-life game. And they have what's called the inspirational shower for scientists.
Wow.
So the other day I had a problem with my code. I'm like, all right, fine, I'm taking a shower. And I came out and I had an idea to fix it. And my daughter goes, oh, you had an inspirational shower, Mom. And I'm like, okay, thanks.
All right, guys. Thank you for joining. So we are going to start officially in the next
1 minute.
less than a minute, actually. So it's difficult to tell everyone good morning or good afternoon or good evening because of the time zone. Sometimes it confuses me, so I prefer to say hello, everyone. Welcome to NOBLEKINMAT's webinar series. My name is Hamza Muhammad Taufik, and our host for today. And I'm also the CEO of NOBLEKINMAT. So I want to officially welcome each and every one of us in few seconds time. So don't take the welcome yet because it's not yet 11 p.m. Korean time. So once it's 11, yeah. So you are welcome. It's 11 p.m. And I would like to officially welcome each and every one of us to this a maiden edition of our first webinar series. Even though last week we have a webinar, which is actually a hands-on screening from EMBL, EBI on Genome browser, and we actually learned how to do a whole lot of things with the Genome browser of EMBL. So it's actually a good traction for us. And briefly, if you are just joining us or you're just hearing about NOBLEKINMAT, NOBLEKINMAT is actually it's a vision, a movement of youths which have keen interest in bioinformatics and the kind of magic that bioinformatics can do. And in case you're wondering what I mean by magic, we have our erudite professor here. So she's going to show you some of those magic. So we are on a mission this 2022 to actually train, empower, and enable at least 1 million people with our great focus in Africa in bioinformatics. So we've kickstarted like a year now, and then we've been doing some kind of preliminary trainings on primer designing and then phylogenetic tree and all of that. So this year we decided to go big, and then we decided to go big by bringing big people. When I mean big people, I mean by bringing Professor Laura and EMBL and more people in the pipeline. So without wasting much of our time, I would like to officially welcome our guest lecturer in the person of Professor Laura Harris, Director of Training, Institute of Cyber-Enabled Research, Michigan State University. Don't worry, we're going to give you a better introduction later, so in case you still wonder who Professor Laura Harris is. So in the meantime, I would like to introduce the brain behind NOBLEKINMAT, which is in the person of Ashraf Akintola, the CEO of NOBLEKINMAT, to quickly give us a brief introduction as regards this webinar, which will serve as an opening remark for this session. Before then, the mode of this webinar, it's for us to actually learn and interact. So please, while our professor is doing the magic, I mean, when she's delivering the webinar on bioinformatics approach, analyzing next generation sequencing, infectious disease and cancer data. So that is the magic we are here to learn today, to see, to gain insights, and then to probably interact. So while she's doing that, I will urge each and every one of us to actually take note of our questions, or we write it down, So after then we can have question and answer session. So we wouldn't like our professors to be interrupted actually. So CEO Ashraf Akintola, over to you, sir.
Thank you so much, Hamza. Thanks for that wonderful introduction. Okay, ladies and gentlemen, it is my pleasure to welcome you all here. As he has announced, my name is Ashraf Akintola Akintayo, and I'm the CEO and founder of NOBLEKINMAT Limited. It is my pleasure to welcome you to this iconic event, one among series of its type by NOBLEKINMAT Limited in Bioinformatics. As our COO has introduced, NOBLEKINMAT Limited is not just a movement, it is an impact development company. And what do we mean by that? We have the mandate of leading a generation of prosperity, of impact and sustainability, especially among African students, researchers, and those willing to learn, and even beyond the borders of Africa, in the area of bioinformatics, data science, health, education, media, and publicity. So we are dedicated to leading the next frontier of Africa's development in all spheres of the continent's transformational plan through the digitalization and democratization of knowledge and essential high-level skills. So we have been doing that for quite a while now, and we are dedicated to building a strong workforce for companies, institutions, and government agencies working towards that collective goal of creating a future for Africa and even beyond Africa. So for more than a year now, my colleagues and I, Dr. Austin Amakiri, who has a PhD in bioinformatics and computational biology from the prestigious University of Liverpool, United Kingdom, and Hamza Muhammad Tawfiq, who just made the introduction now, who graduated top of his class from his master's degree program in applied biosciences from Kyungpook National University, South Korea, one of the best universities in the world also. We have organized online classes to teach bioinformatics skills and techniques to students across the globe. So this year, we said, let us make it more bigger. So the company, NOBLEKINMAT Limited, completed its official registration, and since then, we have set out to create impact, prosperity, and a sustainable development through empowering, engaging, and equipping people at all age grade and levels through the company's platform. So by the end of 2022, we aim to raise and train more than 1 million students, researchers, and enthusiasts, and those who are ready to learn this essentially important high-level skill of bioinformatics across Africa and beyond. And we will fulfill this mandate, God willing, by identifying opportunities at each level of social strata through outreach to government agencies, educational institutions, philanthropic organizations, top researchers, developmental partners, as well as with generous donors and investors to achieve this desired goal. So this is not just a one-time thing. It's a movement, it's a continuous movement. So this seminar is a bold attempt on our way to achieve that goal. And we are so glad that we are and honored to have one of the best and foremost researchers in the field of bioinformatics, especially when it comes to cancer and infectious diseases here with us today. So Professor Laura Harris, our presence here with us today is something we would never take for granted. And in fact, it is the needed morale booster for us to achieve our intended goal. So ladies and gentlemen, at the end of this gathering, I am confident you would benefit immensely and you would get the needed inspiration to forge ahead in your research into cancer studies as well as infectious diseases that have continued to become the #1 cause of mortality and morbidity. So not just in the third world countries, but also in developed nations as well. So once again, I want to thank you for honoring our call, and I wish you and I wish myself a very wonderful and fruitful educative session. So thank you so much, and over to you, Hamza.
Oh, thank you very much for that object-oriented speech, actually. So thank you, CEO, and that's it from our CEO. And from that, we could see it actually looked like an excellent task for us here. when we say we want to train 1 million bioinformaticians, considering the fact that we are also learning, but the most important thing is that we are committed to make impact, and we have really, really important people in the field of bioinformatics here who are willing to go the mile with us. As you can see, evident from the presence of Professor Laura today, which will be talking out to us today about bioinformatics approach, analyzing next generation sequencing, infectious disease and cancer data. So before we start doing the magic, I would like to call on one of our interns to actually introduce the magician of our time to us, in the person of Professor Laura Aris, to introduce our magician. So when I say magician, I'm actually referring to a bioinformatician actually making a lot of impact in terms of diagnosing epigenetics or trying to make a meaningful impact, because we are in a world where it's no longer go to the lab, come out of the lab and all of that. We actually need impact. We actually need a pipelines. We actually need a model that we can use to make
we are in a world
make
where
a kind of inference as regards to what diseases are and the kind of impact they are going to make, and then the predictions, and at the same time to identify biomarkers, to understand therapeutic agents, and also developing drugs, because cancer, it's now becoming what it is actually. So we really need to have magicians like Professor Laura Harris. And then I would like to call on Comfort
agents and also
Shekoni to give us a brief citation as regards the magician of our time,
becoming what
Professor
it is.
Laura Harris.
So we really
Comfort,
need to have
Yes.
magicians like
Yes.
Professor Laura Harris. And then I would like to call on Comfort
agents and also
Shekoni to give us a brief citation
I'm
as
here.
regards the magician of our time.
Okay, thank you, Bars. Thank you for the privilege and opportunity to read the citation of a renowned scientist in the field of bioinformatics. So I wouldn't say in the field of bioinformatics alone, but rather I would say someone that is making mind-blowing research in the scientific field. And that person is not any other person than our humble, beautiful Professor Laura Aris. So everyone, I would like to introduce Professor Laura. Professor Laura is actually an interdisciplinary professional who is so passionate about providing bioinformatics skills to advance healthcare. She obtained a BSc in microbiology degree from Michigan State University in the year 2000, and her first as well as a second master degree in year 2003 and year 2009 from the same institution, Michigan State University. Our first master degree research focused on evaluation of acetohydroxyl acid synthase, assignment to open reading frame, which was actually a comparative study of gene identification models in Methanococcus janashi, while she worked on effect of inflammatory bowel disease on skeletal formation in her second MSc. However, on May 20, 2020, Professor Laura obtained a doctorate of philosophy, PhD in biomedical informatics from Rogers State University in New Jersey, where she conducted a research on genome-wide analysis of NGS data and RNA sequence to identify potential therapeutic target to overcome antibiotic resistance in Staphylococcus aureus. Also, Professor Laura worked at Lansing Community College as an adjunct faculty and also scientific laboratory coordinator. Also, she worked at BioProt Corporation as an associate scientist, and between November 10, 2004 to May 5, 2006, she worked at Pfizer, and we all know Pfizer to be one of the leading industry when it comes to biotechnology and production of vaccine. So she worked at Pfizer as a scientific project associate, where she was responsible for writing preclinical protocols and documents, which includes investigative new drugs, applications, and analyzing experimental data for dermatology compounds. She was She also worked at MBI Lansing as a research associate before she proceeded to the University of Phoenix as an associate faculty and later as a lead faculty area chair. That was between July 1st, year 2018 to February 5th, year 2021. She was at Devonport University as an assistant professor and also scientific laboratory coordinator. And presently, she works at Michigan State University as a director of training for Institute for Cyber-Enabled Research. And that is not all about our humble professor. She has actually contributed immense knowledge in the field of science and bioinformatics, and all of what can be seen, it's evident with her PhD research and as well as more than 6 years independent bioinformatics research. And also she's the founder and principal investigator of ARIS interdisciplinary research, and she has extensive experience designing and maintaining relational databases using Access and SQL. She's proficient in Linux environment, as seen in training over 100 people on the use of high-performance computing clusters since February 2021. At current position as Director of Training for Institute of Cyber-Enabled Research at Michigan State University. She has strong Python scripting ability with machine and deep learning program development, experience evidenced by publicly available GitHub program. So those into text, into, should I say Slack, GitHub knows what GitHub is all about, and she has contributed immensely to that database. That's awesome. Also, Professor Laura has a solid background in pharmaceutical development of a wide variety of therapeutic areas, as evidenced by almost 5 years working with pharmaceutical companies. And now there alone, she has featured in more than 5 articles and also gotten honors in bioinformatics, mentorship, and scientific instructions. And lastly, just to give a little overview of our Professor Laura has authored close to dozens of high impact factor research publications, and she's a member of prestigious scientific organizations such as American Society of Microbiology and also Quantitative Undergraduate Biology Education and Synthesis, QBES, which is an online faculty mentoring network. She has also won lots of grants as a principal investigator and has a wide range of volunteering research experience. So, ladies and gentlemen, it would be a great honor to welcome and present to you Professor Laura Harris. Thank you.
Thank you so much for that introduction. I hope to live up to all of your hype and expectations. And if I knew you were going to read my entire resume, I would have given you a shorter one. But anyway, I'm honored to be here today at this global talk to share with you the work that I've been doing over the last 5 or so years, developing a bioinformatics approach to analyze next generation sequencing data. And I've been focusing that effort primarily on infectious disease and cancer. So again, my name is Laura Harris. If you ever want to reach out to me, you can find me very easily on LinkedIn. I also have my email address and my GitHub right here, and I'll show this again at the end of the presentation. So just to give you an overview of what I will be talking about and how the presentation is meant to go. I'll start with a very brief introduction, and then we have 3 main categories. We will look at gene expression meta-analysis of messenger RNA expression data, and this is specifically looking at SARS infection in lungs. We will then transition into looking at pathway activity meta-analysis of the same messenger RNA expression data, except in this case we're looking at antibiotic resistance in Staphylococcus aureus. Then we're going to switch gears a little bit and look at cancer. And we're going to look at methylation site meta-analysis of DNA methylation profiling data. And this is going to be examined across various cancer types, 12 if I remember correctly. Each of these three categories is broken down into the same general flow or subcategories, where I'll introduce a little bit of the background and significance of the particular topic. We will then identify a panel of either genes, pathways, or methylation sites. We will then verify the panels that we identify. And then we will take those panels and compare them across various signatures of genes, pathways, or methylation sites across cancers, antibiotic resistances, or SARS infections. And the fun part of this presentation, at least for me, is that we will do all of this using effectively the same computational approach, which is freely available to you on my GitHub page. So let's get started. Oh, and one more thing. I do intend to pause briefly for questions between each category, so you don't have to hold all your SARS questions till the very end. So my overall goal for all of my research is to elucidate molecular changes associated with disease mechanisms. And I'm going to do this using computational analysis of next-generation sequencing data that is publicly available in knowledge bases such as Gene Expression Omnibus or the Cancer Genome Atlas. And I'm going to use this next-generation sequencing data to identify potential targets for therapeutic development to overcome those diseases. This project is the first time a meta-analysis approach of gene, pathway, or methylation signatures has been used to identify molecular changes associated with infectious disease and cancer. And I'll define what a signature is in just a moment. So, being a somewhat ignorant American, I did my homework before coming here and decided to look at what diseases are predominant in Africa. And so thanks to the World Health Organization, they had leading causes of death in low-income countries, such as, for example, Somalia or Madagascar. And then there's leading causes of death in upper middle class income countries. So these are going to represent causes of death from places like South Africa or Nigeria. And what you'll notice is that you end up with a bunch of communicable diseases such as lower respiratory infection, neonatal condition, tuberculosis, malaria, are still very prevalent in lower income countries. But as countries develop in economic status, they start to get at control over infectious disease, and they start to have issues with cancer, heart disease, and stroke. And then if you get into more economically rich countries, like, for example, the United States, where I'm from right now, you get way less infectious disease. Now, with that said, notice I'm doing 2019. That's because the World Health Organization has been overwhelmed with COVID-19. And so keep in mind that COVID-19 is now one of the top killers in all countries, regardless of economic status. Now, the black arrows I have here represent the topics that we will be covering today. So as you see, I've already been able to apply my computational approach to quite a few of the diseases that are haunting humanity today. So then let's get started with our first example. We're going to look at gene expression meta-analysis of messenger RNA data, and we're looking specifically across SARS infections in lung cultures. If you want to read more detail about this study I'm about to talk about, it is a published study in the Frontiers of, I believe, Immunology from last year. So just a little bit about SARS viruses, in case you're not a virologist, and I was not until 2 years ago. The SARS family comes from the beta coronavirus family. And these are viruses that are usually not life-threatening. They're usually the common cold. However, 3 times in human history, we have noted deadly strains of beta coronavirus. The first time we saw this was back in around 2001 with a severe acute respiratory syndrome we called SARS. This was caused by SARS-CoV-1. A few years later, around Saudi Arabia, we had the Middle East respiratory syndrome, or MERS. This was caused by MERS-CoV. And then, of course, everyone is now all too familiar with COVID-19, which is caused by the SARS-CoV-2 virus, and fortunately now is becoming an endemic rather than a pandemic. It's taken a while to get there. So the SARS-CoV-1 and the MERS outbreaks did not become pandemic because we were able to use aggressive infection control to contain them. However, COVID-19 became a pandemic. Fortunately, vaccines have helped in terms of reducing the spread, even though we see strains like Omicron and its variants coming up. And there's still limited treatment options for all SARS infections. And there's a huge concern that SARS is going to continue to mutate into SARS-CoV-3, which may become another pandemic in just a few short years. So just to make sure we're all on the same page, we understand, what is gene expression? So a gene is a piece of DNA that is the basic physical and functional unit of heredity. We know that genes are stored in our DNA, which is in the nucleus of a cell. A gene then goes through transcription to make a messenger RNA or a mRNA copy, which then leaves the nucleus and then undergoes translation to form proteins, which effectively are the building blocks and the functional units of a cell. So when I say mRNA gene expression, I really mean the process of producing a protein product from a gene's instructions. So turning the ATCs in genes into a functional protein. And to do this, or to evaluate it inside a cell, I'm going to use messenger RNA expression data from public knowledge bases. And here is how that data is generated. So we're going to take an RNA sample, say from a SARS-infected cell, and we're going to take RNA sample from a normal cell, maybe a mock-infected cell, and we're going to convert those RNA into complementary DNA, or cDNA, using a reverse transcriptase from HIV. We're then going to fluorescently tag the samples based on where they came from, and we're going to hybridize them to a microarray or an RNA-seq chip to then look at the fluorescence to see which probe is more dominant. This then gives you gene expression data for a whole profile of up to or over 50,000 genes at one time. So that data looks like this, and this is just a snippet. I mean, it goes 50,000 down and a few hundred across. You kind of need a decent computer for this. But anyway, we're going to have our genes listed, and we're going to have our samples listed, and some samples are going to be experimental, in this case SARS-infected. Some samples will be control, in this case mock-infected. And I'm going to do a basic statistic called a t-score, or a t-test, that basically compares the mean of each population to determine if there's a statistically significant difference. I calculate a t-score for each gene, and then I can rank the genes based on that t-score, from high expression in the experiment versus the control, to low expression between the experiment and control. And in the middle of my signature, we would see effectively no change, or a 0 t-score. So then this gene signature that I can define out of these 50,000 or so genes is a list of genes ranked on that differential expression. And in this case, I'm using a t-score. So then I can go find data sets in Gene Expression Omnibus. And in this case, I find two data sets that are comparing Petri dish samples of SARS-CoV-1 versus mock. And so I can create a signature, just like I showed you on the prior slide, and I can do this for each individual data set that I find. I then can take the top 500 genes of the positive tail, so these are the overexpressed genes in SARS versus mock, and I can take the bottom 500 genes, the negative tail, so these are genes that are higher expressed in mock compared to SARS, and I can use those as query gene sets to compare them using gene set enrichment analysis against a reference signature from another data set. So doing this then for the positive tail and the negative tail, I get these enrichment plots that you see here in the middle. And so my enrichment score is going to go up every time a gene from my query set, in this case my positive tail on top, matches where in the reference signature it's located. And you can see a lot of my positive tail are located towards the red positive end of my reference signature. And the same thing for the opposite blue negative tail. And you'll notice this normalized enrichment score has a significant p-value. So this says that my query set is significantly enriched in the positive or negative tail of my reference signature. So what you'll notice is now that I know that effectively my two signatures are similar to each other, I can then figure out which genes in the tails are similar to where they align on the reference signature. And so you'll notice there's a maximum enrichment score that gene set enrichment analysis calculates. All the genes that go into making that maximum enrichment score are called leading edge genes. These are the genes that are thought to be the most significant in terms of biological relevance. So I'm going to take leading edge genes from both my analysis, the positive tail and the negative tail, and I'm going to define a positive gene panel of around 233 genes and a negative panel around 114 genes. Now that I have these panels of genes that represent a SARS-CoV-1 infection, I can look at those panels and see what genes are in there. Identify genes from membership. I can verify the genes in those panels by comparing them to other data sets. And I can compare them to completely different strains of SARS, like SARS-CoV-2. So let's do that. Let's first see what's in those panels I just identified. Well, out of the positive panel, 13 of those genes, or around 5.5%, were already reported in the literature. So that tells you that the computational approach that we just showed can predict genes that have laboratory established association. However, 5% of the panel is quite small compared to what the computer identified. So there's a lot of genes in there that could be associated with SARS and could be potential targets. Some of those include zinc fingers and solute carrier family proteins. In the negative panel, no genes had previous associations that were reported. But again, we see zinc finger and solute carrier family proteins popping up. So now that we have those panels, and we've looked into those panels to see what genes are there, let's compare those panels to other data sets using a similar laboratory approach to generate their data. So verification. So I'm going to take my panels here, and I'm going to use them as query sets for gene set enrichment analysis, and I'm going to do that against 4 independent data sets. One of them came from the same cell culture, the human airway epithelial cell cultures that the panels came from. So this would be called a true verification data set. All experimental parameters were the same as my original data sets. However, there are these other 3 data sets that are exactly the same experimentally, except they use human adenocarcinoma cell cultures instead of human airway epithelial cell cultures. Both of them are lung cultures. They come from lung cells, but they're different types of lung cells. So while we don't expect to see a difference, there could be a subtle difference that the computer's not picking up, etc. So these are not completely true verification data sets like this other one on the side is. So this is just the one data set looking at epithelial cells. This is the true verification data set. And you'll notice when you compare the positive panel, you get a positive enrichment that is statistically significant. You compare the negative panel, you get a negative enrichment that is statistically significant. Now I wanted to know, will these statistically significant numbers occur randomly? Is the signal I'm seeing here true? And to do this, I developed a random model. So I took 233 genes out of the 5,000 that are, 50,000 that are there, and I took 233 at random. And I did this 1,000 times. And I just reran gene set enrichment analysis, and I collected the enrichment scores. And you'll see them plotted as a distribution plot here. I did the same thing with the negative panel as well. Took 114 genes, 1,000 times randomly, and plotted the distribution. What you notice here is that my panels were able to achieve normalized enrichment scores well outside the normalized enrichment scores that were randomly generated. So this tells me that even in an independent data set, my panels are better, statistically significantly better than random chance. So this is really good. Support that these genes are associated with a SARS infection. So then let's look at those other not completely true data sets, in the ones in the adenocarcinoma cells. Again, positive panel, statistically significant overexpression, statistically significant. You see where I'm going with this. In every case, we see that the panels are able to predict the SARS infection better than random chance. So strong validation or verification, I mean to say, that the panels identified are associated with a SARS infection. So then let's look at the leading edges across all those data sets. What genes are identified as contributing the most each time? Which genes are the most consistent? And when we do that, we find 55 of the positive panel genes and 22 of the negative panel genes are consistent across all 4 non-overlapping data sets. 10 of those 51 genes were previously reported in SARS infection, but in the negative panel, they are not. So that leaves around 53 genes that have no prior association with SARS that our computer predict and could make interesting potential targets. So let's move out of the Petri dish into an in vivo model. And at this point in time, we were doing this like couple weeks, maybe a month after the pandemic started for COVID-19. So there wasn't a lot of data available in humans. And we're still dealing with the SARS-CoV-1 in this study. So again, not a lot of data in humans. So I went for mice. And so these are mice that have been infected with SARS, and then comparing the gene expression between the mice. The positive panel is still statistically significant, but the negative panel now loses its significance. And then you can see when you do the random model here that the positive panel is still at the top end of what can be generated randomly, not so much for the negative panel. So this tells us that the positive panel still is enriched and associated with SARS in an animal model. But the negative panel probably is some sort of a signal specific to the Petri dish. So then looking at what genes overlap between the Petri dish and the mouse, I do a Venn diagram. And so what you'll see here on the left side is these are all the leading edge genes in the human. And then in the dotted circles here, you have leading edge genes for the mouse. And you're looking for the overlap between the dotted circles and the solid circles. Now, we wanted to acknowledge the fact that some genes are present in human, but not in mouse. And other genes are present in mouse, but not in human. So if we identify a gene that's in human, we did not want to exclude it because it wasn't present in the mouse. So we did consider, and we found 2 genes this applied to, where we identify them, but we now lose their signal simply because we're changing between in vitro and in vivo. Okay. So total, we have 20 genes total. And here they are. You'll see them listed across the top here. And what I've got here are the signatures we used, the 2 for identification, the 4 for verification, and the 1 with the mouse at the bottom. You'll see whether or not a gene was statistically significant by t-score. And you'll notice the black boxes mean the gene was not present in that signature. Now, what this shows you is that some signatures do not have t-score significant genes. But because we take the 500 top genes regardless of their t-score significance, we're able to detect genes that are borderline statistically significant. They may not have enough math to support them being fully statistically significant, but they still have biological relevance. And we want to consider them because they do have that biological relevance. And this method allows you to do that. And so that's why these gaps in the t-score heat maps are important. It shows you genes that would have been thrown out by other analyses that are being picked up by mine. And so now we've looked extensively at the SARS-CoV-1 IC-SARS strain. We looked at it in humans, we looked at it at mice. Now let's compare that to other SARS-CoV-1 strains, and eventually in MERS and SARS-CoV-2. So here we have some other fully virulent strains. There's Urbani, and then there's this MA15 strain, which is a mouse-adapted Urbani. These are all fully virulent strains. And in the case of the mouse, we actually do varying inoculation doses. Then we have a couple attenuated strains. These are ones that were developed for vaccine production. They're weak, and some of them have particular gene deletions that you see noted here. And so now we're going to take those panels that we identified for the IC SARS, and we're going to compare them across all the strains. Let's see what we find. So it's a little hard to see because of all of the signatures, but what you'll notice is consistent white stars across the positive panel. So it didn't matter if we were looking at these infections in humans or in mice, as you see on the right side here. We get statistically significant enrichment of the positive panel across all of them. And we also see it regardless of inoculation dose. When we look at the random model on the right side here, you'll see the little red triangles. And those are where our positive panel is statistically enriched based on random modeling. So again, these genes in the positive panel seem to be consistent and associated with a SARS-1 infection across 7 different strains. Looking at the genes from each strain to try to figure out which ones are in common, we find 5 that are common across all 7 SARS-CoV-1 strains. These are the 5 genes. Here is the T-score heat map, like I showed you a few slides ago. And again, you'll notice sometimes genes are not included in a signature, and sometimes they're not picked up by a T-score significant test. But our approach is able to find them. And what's interesting about these 5 genes is that they are all related to interferon. So we'll talk about interferon and what that does in a SARS infection in a moment. So now that we see these 5 genes for the SARS-CoV-1, let's look at MERS. And when we, oh wait, hold on. Yeah, this is MERS, I'm good. So when we look at MERS and SARS-2, we see the same thing in terms of our positive panel is statistically enriched across all these signatures, with one exception, and that's this one right here, this GSE 155518. This one messed with me because you see I have this beautiful signal across all these strains and all these doses and mouse and human, and what's up with this one study? So I actually emailed the people that generated that data for Gene Expression Omnibus, and they said, we didn't make the RNA right. So we would disregard those results. So just something to keep in mind, if you do see those anomalies, go back to how the RNA was made, because how the RNA is made and the quality of the RNA that's analyzed impacts the results of any bioinformatic analysis. So anyway, we still see statistically significant enrichment in non-random. Good to go. Now I'm looking across the panels and I still see my 5 genes, usually statistically significant by T-score, except for our favorite little gene data set down here. And so these 5 overexpressed interferon genes were found across all of the gene signatures that we found, except for the one we had to throw out. This suggests that targeting interferon response might improve patient outcomes when people have a SARS infection. And we see reported in the literature that interferon is a huge driver for patient mortality and whether or not they're going to develop a severe inflammatory response that might kill them. We also find it in the literature reported specifically for other cell types. So, you know, you're looking at the human airway epithelial against SARS-CoV-2 is associated with interferon therapy. Interferon therapy has been successful in treating some COVID patients, though sometimes the timing and the route of administration is debated. But the point is, using a gene set enrichment analysis based meta-analysis approach can identify genes that can be targeted to overcome a SARS infection. And so with that, I'll take just a moment and I'll pop out of here and ask for a couple questions. I'll take like 2 questions on this right now, just for time's sake. Hopefully someone's been monitoring the chat.
Yes, we have been monitoring the chats.
Yes.
Are there any questions, or should I just continue?
I have 2 questions actually.
Please. Great.
Yeah, that was really a very nice presentation. So my first question starts with T-score. Why T-score? I know that in calculating differential expression within genes or within samples, there are a number of different tests available, including the T-score. But some people have different preferences. So do you have a specific reason why you use T-score instead of the others?
Okay. There's 2 main reasons I prefer T-score. One is because it's better for larger sample sizes. I find that with T-score, if you have sample sizes that are 3 or more, it just tends to give you a more robust signature. It's easier to reproduce. I've played around with fold change and variations, log 2 fold change, etc. And I've played around with Z-score. They're good for small sample sizes, like if I'm doing a single cell analysis or I only have 2 samples, but it starts to get flimsy and your signal is not as reproducible. In the future, I do want to conduct a study that looks at those effects, and the literature has reported there are substantial effects about the metric you use to rank versus the outcome of gene set enrichment analysis, but it seems to be very specific to the approach. So my short answer to that would be it works best.
Okay, that's fine. Then my other question is, so listening to your presentation, you have slightly different number for your positive panel and slightly different number for the negative panel.
Yep.
And I was thinking that to get a similar distribution, you want to have a similar number of panels. So if I were looking at 250 genes, I could use 250 positive and 250 negative. Or I could use maybe 250 and 125, but I have to normalize the effects. So to make it like to make it on par with one another.
Yeah, yeah.
I don't know.
I see where you're going with that. And where your misunderstanding is the fact that we're not using one signature to define our gene panels. We're using 2 and comparing them. So your approach of taking 250 genes, we actually did at the very first stage. Remember, we took 500 and 500? Mm-hmm. But then taking those, we whittled it down to the 233 and the 114. And we do that because those are going to have the most biological significance. And then we found that it doesn't matter for a random model if you pick the numbers 233 or 114, the distribution of the random normalization score doesn't change that much, as you see on the one slide. So we find that once you whittle it down, through that comparison between the 2 identification signatures, all of that becomes normalized, if you will.
Right. Thank you. And I think my final question, and then I'll go sit down and listen to others, is so when you did the gene set enrichment analysis, you have the potential to set your background distribution.
Yes.
So I know you did mention that they were positive or you got significant enrichment, but I didn't hear you mention what background distribution you used to compare the samples or the panels. So.
Yeah. So I'll just flip back to one of them. This will work. So basically, you've got the reference signature. I don't know if you can see my cursor actually moving. Hopefully you can.
Yeah, I can.
Perfect. So you have the reference signature here, and this is, 50,000-ish genes all lined up by their T-score. So I'm only taking 114 or, 233 at random. And then that would be my new panel. So the null distribution comes from the reference signature. You're just randomly picking from it 1,000 times. Does that make sense?
Yeah. That is super cool now. Thank you. Yeah.
Perfect. I will be happy to answer more questions about SARS at the end of the presentation, but I still do want to get through Staphylococcus and cancer, or we'll be here all day. So if that's okay, I'm going to proceed on.
We'll keep the questions and we'll ask them after the presentation.
Perfect. So I'll take another mini question break right after staff, and then we'll move on. Okay, cool. So now, if you want to see the continuation of the SARS story, because we do have a second paper published on SARS that looks at pathway activity, which is what I'll start discussing now. You can go look that up. But I'm actually going to reach backward to my dissertation, the thing that kind of started this all. And I'm doing that because I'm talking to a population specifically focused on Africa. And infectious disease for antibiotic resistance, particularly Staphylococcus aureus, is like your biggest killer for neonatal infections. So it seemed kind of erroneous of me to just continue on the SARS story without introducing this. So the same approach I'm going to show you here for Staphylococcus aureus was also applied to SARS. Feel free to go look that up and read it on your own. But here we're going to focus on the pathway activity meta-analysis using mRNA expression data to look at antibiotic resistance in Staphylococcus aureus. And this paper was published by Rutgers because it's my dissertation, so it can be found here. So for those, again, that aren't bacteriologists, I was classically trained as one. What is Staphylococcus aureus? So Staph aureus is a bacteria that can cause life-threatening infections. It is part of your normal skin flora. And about 50% of the population, human population, is walking around with an antibiotic-resistant staph species on their skin. Doesn't usually cause a problem until you're immunocompromised. So when you go in for things like surgery or you have an accident, and we then end up with an infection that is untreatable. So when you have those infections, the common treatments are antibiotics, penicillin derivatives like methicillin or oxacillin, or vancomycin. These are all cell wall synthesis inhibitors. You can have cell membrane disruptors like daptomycin, or you can have protein synthesis inhibitors like linezolid. But resistances to all of these antibiotics have developed, and we now have superbugs that are resistant to every antibiotic humanity has. And this is not just a third world Africa country issue, though they are the ones getting hit the hardest. This is a worldwide crisis that is hitting developed countries. And Staphylococcus aureus is the most common cause of late onset infection that's leading to significant mortality, morbidity, and prolonged hospitalization worldwide. So what is pathway activity? Well, a pathway is a series of molecular actions in a cell, usually made up of individual genes that work together in kind of a process to make some sort of a product or a phenotypic change. So the example that I have here is aspartate to lysine biosynthesis, where you start out with an aspartate, that aspartate binds to an aspartate kinase, goes through a series of changes, and then ends up producing a lysine. However, that lysine can attach itself to the allosteric site of the aspartate kinase in order to cause a conformation change and shut the pathway off. So it's a self-regulating pathway. Aspartate has to be present in order to move the pathway forward, but if enough lysine is present, we can shut it off, even if aspartate is present. So we'll revisit this in a minute. But when I say pathway activity changes, I'm specifically referring to a measure of expression changes, gene expression changes, for several genes in a pathway, several genes working together to accomplish a cellular function. So I can do that using a very similar approach to the one I took with gene expression in SARS. I can get some messenger RNA expression data from the public repository. And in this case, I'm going to take some data that has resistant bacteria and sensitive bacteria. Now we're simply going to start untreated to look at gene expression without treatment. But we will advance when we get into comparison to look at treating it also. So anyway, I take my resistant and my sensitive, rank them by T-score, get a gene signature. So this is exactly what we saw before. But now, to make the pathway signature, I can take the gene signature that I created and compare it to pathway gene sets. So these are query sets, lists of genes that are related to a pathway that I can get from public knowledge bases like KEGG, Gene Ontology, Patrick. And you download these pathway gene lists, use them as query sets, and then you can rank the pathways based on normalized enrichment score so that every pathway has its own individual normalized enrichment score. So in this way, I effectively use gene set enrichment analysis twice. Once to make my gene signatures, and get my gene panels, and then to take those signatures and make pathway panels. And so pathway signatures are a list of pathways ranked by normalized enrichment scores. And we can do the same thing. We can discover pathways in them, use them for panel verification and comparison. So in this case, I started with VRAR-SR-driven resistance mechanisms. So let me back the train up a minute. VRAR-SR is a membrane transporter for vancomycin. And the literature shows that if you mess around with this VRAR-SR gene, you then can change resistance levels. So on one side, I have a vancomycin and daptomycin resistant strain that has high VRAR function. And on the other side, I have a sensitive parental strain that has normal VRAR function. We need VRAR function or otherwise we're not transporting. So we do need some baseline, but too much means resistance. And remember, these are untreated cultures. I just want to see how the bacteria reacts. Now, in this case, there are not very many expression data sets available. So I could not do that comparison to get a gene panel. So I actually had to take the approach that was questioned in the Q&A a minute ago, where I just take the top 20 pathways and the bottom 20 pathways. And this captures about 25% of the total pathways. So then if I match my ends, I'm pretty confident that I'm going to have signature matching. So I'm just going to define my panels with that, 20 and 20. simple. When I when I look at those 20 pathways, I notice that I find histidine and I find aminoacetyl tRNA biosynthesis, and I find succinate dehydrogenase. These 3 pathways were already identified by the gene level analysis that the published paper that made my data set found. But what they did not find that I found was that lysine biosynthesis was statistically enriched. And therefore, lysine biosynthesis seems to play a role in vancomycin and daptomycin resistance. And this is new to the literature that was not reported before. So can we verify that in independent data sets? The short answer is, of course we can. But remember, it was hard to find data sets. There's not a lot of people doing these experiments. So I had to switch genes a little bit. So VRAR actually works alongside GRAR, V and G. And so the 2 of them work together to do the same cellular effect, to create vancomycin and daptomycin resistance. So I use GRAR as my verification because I could not find any VRARs. I'm hoping I'm pronouncing my V and my G correct so you understand what I'm saying. So that's why you see GRAR instead of VRAR. Best you can do. And so here, we go ahead, we're going to rank by our signature by fold change like I talked about in the Q&A. Down here is T-score. But again, we're making pathway signatures just like we did before. We're going to compare the pathway panels, the 20 and 20, from VRAR to the GRAR signatures. And what you'll notice is my positive panels are statistically significant. And when I look at the random model, they still hold. However, the negative panel only is statistically enriched for one of my signatures, but not the other one. And we see the same thing in random model. So just like the SARS, it seems like the positive tail is more reproducible than the negative tail. Now, don't think it's something about my procedure, my algorithm, because I'll prove otherwise with cancer. But anyway, this is just how the data is flowing. So I'm going to focus on that positive panel. And when I look at that, I see my histidine and my succinate dehydrogenase. Okay, so I'm looking across. Histidine is reproducible. Okay, it was reported. The aminoacetyl tRNA biosynthesis is not consistently reported. Succinate dehydrogenase, not consistently reported. Lysine biosynthesis, look at all the reds. Consistent across. Looks as great as histidine. So all 3 data sets share increases in histidine and lysine. Histidine was reported. Lysine is new. So as we move into the comparison to other antibiotics and other data sets, I want to make sure we're all clear on what our definitions are. So up till now, I've been discussing what's called mutation-driven resistance, comparing resistant and sensitive bacteria under effectively the same treatment conditions. No treatment, or in one case, we treated them lightly with colostrin. It was only used as a stimulant for the gene that we were expressing. Moving forward, I'm going to be looking at 2 different types of data sets. There's antibiotic susceptibility data sets, where there is a difference in treatment response between resistant and sensitive. So in this case, they are treated with vancomycin or linezolid. But now one is resistant and one is not, and you see what the response to that treatment is. Similarly, there's antibiotic response. These are sensitive strains. that are challenged with just sublethal doses of antibiotic to see how the expression changes under challenge. And the idea here is that the under challenge sensitive, the antibiotic response group, is the precursor to the antibiotic susceptibility group. Cool. So let's take a look at this. I was able to find 2 signatures that look at vancomycin susceptibility. I was able to define 2 signatures that looked at oxacillin susceptibility. And I found 4 signatures that I could define looking at linezolid susceptibility. Now remember, vancomycin and oxacillin are cell wall disruptors. Linezolid is a protein synthesis disruptor. Completely different mechanisms. I'm also able to define antibiotic response between vancomycin, fluoxacillin, which is similar to oxacillin, and linezolid. Okay. And I'm going to use all these signatures as references against those pathway panels that I identified earlier. You'll notice a lot fewer stars here. And part of that has to do with the idea that they're different mechanisms. So, for example, when we're looking at the linezolid susceptibility, our positive panel becomes blue. It actually starts heading towards the opposite end. Well, they're different mechanisms, so we don't expect them to have similar enrichment. But that doesn't mean we cannot find pathway similarities across them. So as you start looking through here, okay, I can see vancomycin is up, and then of course when I look at the down panel, it's pretty down, but then my panel was defined by a vancomycin resistant organism, so it's not surprising I get a match there. I start to see a reversal with the oxacillin and linezolid, where my positive panel starts getting blue, my negative panel starts getting red. Okay, so these are all things to consider when we look at the biology. And at the bottom here, you've got your random models, so you can see how they compare. I won't spend too much more time on this, because the real take-home story becomes this, and moving forward to the next slide. So what pathways do we see across all of these signatures? And so you'll notice, again, not very many stars up here, which means that my GSCA-based approach is able to pick up things that would be missed if I used traditional Fisher's exact test scores, etc. But if you look at the relative frequency and leading edge, you'll notice that in green, the highest leading edge frequencies are lysine pathways. And that becomes interesting because remember, lysine was not reported but was consistent in those mutation-driven signatures. So we decided to finalize all of these predictions by going to the wet bench. This is the only lab experiment that I'm actually done that I'm going to show you. Everything else has been done by somebody else in the lab. I just play with their data. But anyway, so because we keep seeing lysine biosynthesis, I ran what's called a disk diffusion experiment. So I took some petri dishes that had tryptic soy agar in them, and I put either water, that's my vehicle, lysine, aspartate, or phenylalanine. I chose phenylalanine because it was a pathway that was in the middle of my signature. It didn't change between antibiotic resistance or sensitive. So I should not see a change in it when I look at it in the lab. It's my negative control. And so I'm going to put lysine or amino acid onto the plate. And then I'm going to stamp onto the plate antibiotic discs. One for vancomycin, one for oxacillin, one for linezolid. And I'm going to put it in the incubator and I let it grow. And I come back and I measure what's called the zone of inhibition. So the zone of inhibition is a clearing of no growth around the disc, where the antibiotic was successful in killing the bacteria. If I have no or a very small zone, that means my organism is resistant. If I have a large zone, that means my organism is sensitive. A lot of organism died from that antibiotic to make the large zone. So I'm looking for size of zone changes. And that's what C over here shows you. Vehicle is pretty similar to phenylalanine as we go through. So water and phenylalanine do not change the size of my zone. But you'll notice that lysine does statistically change the size of my zone with relation to vancomycin. Aspartate changes the size of the zone for linezolid. And oxacillin changes the size of the zone regardless of if I give it lysine or aspartate. So while I do not know the exact mechanism that's driving this observation, the computer predicted lysine biosynthesis was related to antibiotic sensitivity, and the zone of inhibition size changes confirm what the computer predicted. Now, further experiments are needed to figure out why this is, what lysine, what role lysine plays in antibiotic sensitivity, but there clearly is a role. So overall, for this project, I was able to identify and verify pathway activity changes associated with mutation-driven resistance, specifically increases in histidine and lysine through the VRAR and GRAR mechanisms. I then compared those identified pathway activity changes to vancomycin, oxacillin, and linezolid susceptibility and response changes, and find that lysine biosynthesis is consistent across all those antibiotics. So it could be a global resistance mechanism for staph. And then, of course, we took it into the lab, did the disk diffusion, and confirmed those predictions. Okay. So at this point, again, I'm going to stop for just a couple of questions, and then we will move on to cancer. I do want to get done before 11, at least my time, so 45 more minutes. So at this point, again, I'm going to stop for just a couple.
Thank you so much, Professor.
And then we will move on.
Hamza, are you there?
I do want to get done before 11, at least my time.
Yes, please. I'm here.
Okay.
So thank you so much, Professor.
I think there are some questions that
we have some. Yes, please. I'm here. Yeah, so there's a question by Marcelena Benjamin that you should please explain the verification of GSEA plots by random selected genes.
Sure. We can do this from either aspect. Where do I want to go from this? I'll go back to SARS just because I like the distribution better. Okay, so here we are. And it's the same approach whether I'm showing it as a distribution or as a box whiskers plot. Same concept. Okay, so what we've done is we've taken the particular reference signature and we've randomly taken out of it panels that are the same size as the panels we are testing. So if I'm dealing with the SARS example, I'm taking panels that are, in the hundreds. If I was dealing with the pathways, I'm taking panels that are size 20. But the idea is the same. You're going to pull random pathways or genes to make a panel, and then rerun gene set enrichment analysis to get a normalized enrichment score. And you do that 1,000 times. So then here, what you see is 1,000 normalized enrichment scores randomly generated. The triangles are the actual panels. So this is what I'm actually able to achieve and where it's located based on 1,000 randomly generated panels. So the p-value here is what's called a null distribution p-value. So you'll see like this one right here has 2 stars. That tells you p-value less than 0.01. So that means out of 10, or how do I put it, out of 1,000 trials, 10 of them are equal or better than the one my panel was able to achieve, which is what you're seeing on the left side. I hope that answered the question for whoever was asking.
Yeah, I believe so, Professor. We have some couple of questions, or more like requests.
Yeah.
That if it's possible, that's the slide, the presentation looks more theoretical, and they're kind of interested in learning how to do this practically.
Mm-hmm. Okay, so I will actually address that question at the end of the presentation because I do have that as part of the conclusion.
Okay, that's quite impressive, yeah. Professor. So I think that's all the questions so far from the chats.
Okay.
Don't know if anyone has any question, then maybe we'll proceed to the next category.
There's another question, actually.
Yes, please. Go ahead.
For miscellaneous question, I think the question was that they want to know how to genome browser. So can you show us how to mine some genome browsers and how to do this practically, like genome browsers?
Okay, this is a catch-22. So everyone wants a simple way to data mine the repositories, but there is none. I literally spend days combing through Gene Expression Omnibus in order to find the data sets to use for these studies.
Wow.
So I will be putting on my GitHub in a couple of weeks a video and a template on how to do it, but there is no easy way. And it's time consuming, it's a pain in your butt. Someone needs to figure out a better way we could do this, but I feel ya. And the GitHub will have something around that in the future, but not immediately.
All right, Professor. Thank you so much.
Okay. So I'll go ahead and cut questions for now. I will double back to the GitHub at the end, I promise. And then the last little section that I have is on cancer. And so this is kind of interesting because up till now we've been dealing with messenger RNA expression data. But there are other types of next sequence generation data. And so one of the questions I had with this particular project is, can we use this similar approach on methylation data? And we actually see that we can. Now, this paper is fresh. This is actually the first time that I'm presenting this, so consider yourselves lucky. The paper is in review by PLOS, so we're waiting to hear back. But right now you're my peers, so please review me. All right, so this is the methylation site meta-analysis of DNA methylation profiling data, and this is across various cancer types. So cancer. Cancer is a disease in which some parts of the body, their cells start to grow uncontrollably and start forming tumors. If those tumors break apart, then the cancer cells can spread to other parts of the body. That's called metastases. Regardless, you need to be able to control your cell growth or you will start growing in all sorts of places you should not be growing, and that will eventually cause death. Cancer is the leading cause of death worldwide. There's 10 million or so deaths from cancer in 2020 alone. And I understand from the World Health Organization about 6% of those deaths come from Africa. Nearly 1 in 6 deaths globally are from cancer. And as we showed really early in this presentation, as those developing countries in Africa become more economically secure, cancer will become a bigger problem than infectious disease. Lung cancer is the leading cause of cancer death, and lung cancer causes around 25% of all cancer deaths. Lung cancer kills more people every year than colon, breast, and prostate cancer combined. And almost all cancer cells, regardless of what type, seem to possess multiple epigenetic abnormalities. And these epigenetic abnormalities are directly related to that cell going cancerous. So this is part of why we were motivated to look at methylation in cancer, because it's a huge part of the disease process. So what is DNA methylation? Well, methylation is an epigenetic mechanism that uses histones to control cellular gene expression. So effectively, it's a way that the cell can turn gene expression on and off based on moving these proteins, these histones, to particular locations. So I've got this picture I took from the Moore paper. And you'll see here you've got methylation, which are these black dots, and then you've got white dots. These are unmethylated. When you're methylated, you can move. And when you move, your gene can express. But if you're black, you're kind of stuck there. And then the gene underneath you cannot express. And that is usually a challenge, because if I'm blocking genes like a tumor apoptosis gene, you know, like a cellular apoptosis gene, now my cell's going to go tumor because it doesn't have the ability to express apoptotic genes. So we want to have a good methylation pattern that isn't too hypermethylated, so it's going to shut genes off when they're not supposed to be. But we also want enough methylation to keep the genome stable, or otherwise the DNA starts falling apart. So when I'm saying methylation changes, what I'm really saying is a measure of methylated cytosines between cancer and normal samples. Okay. So how is methylation profiling data usually generated? Well, there are some older ways you can find, but nowadays it's usually done by this bisulfite conversion. So basically what happens is you treat your DNA sample with a bisulfite, and that's going to turn any unmethylated cytosines into uracils. Any methylated cytosines will still be cytosine. This bisulfite conversion can then give you the ability to see whether or not a particular site, genome site, is methylated or not. And then you can compare those methylation patterns between cancerous and non-cancerous. So we do our conversion, we're going to make a bunch of copies through PCR, we're going to attach on our fluorescent probes, because remember, we're going to need to take that picture, select out proper size, get your image, do your analysis. So methylation profiling data is basically a comprehensive profile of the methylation patterns for 50,000-ish genome sites. The data looks remarkably similar. So I have a list of sites, and I have my samples. Some are cancer samples, some are non-cancer samples. And I can do my good old t-score, or my fold change, or whatever ranking metric I want to use. And so in this case, again, I'm using t-score, and I can make a methylation signature. 1 second. Let me grab something to drink real quick. This is what I get for talking for over an hour. Okay, so methylation signature, list of genomic sites. that differential methylation pattern. Now that I have methylation signatures, and in this case I'm going to use lung cancer adenocarcinoma samples. These are patient samples, by the way, they're not Petri dish samples. I can then go ahead and compare my lung adenocarcinoma to my adjacent tissue samples and create my signatures. Take my top 500 probes from each end for my query, compare them, get leading edge. Now, keep in mind that this is my most recent study. So I started with that Staphylococcus aureus study where I just took 20. Couldn't do anything else. Then we moved into SARS where I had more data, so I would take 500 and then combine take the 500 and take their leading edge genes. In this case, I'm going to do it twice. I'm trying to make identification more robust. And so in this case, I'll do it once the same way we saw with SARS, but then I'm going to do it a second time where I flip, inverse my signatures. And this then gives me a second positive leading edge and a second negative leading edge from the same two signatures. When I compare the positive leading edges and the negative leading edges, now I can get panels that are a bit larger. So you see how now I've got 320, 304. I get more consistency, I get larger panels. It's a more robust process, okay? But everything in science, tweak it a little, play with it. Does it work? Does it not work? Well, we're still evolving too. So out of the 320 probe panels for the lung adenocarcinoma methylation panel, most of the probes already had identified associations with lung cancer. Quite a few of them, almost 30 probes, were associated with the Hox genes. 11 probes, though, had no previous association. So again, we're back to the program is able to pick up things that humanity already knew and contribute some additional information. And the same thing occurs with our negative. We have some probes that were already identified, like TNF receptor-mediated apoptosis is down in methylation. But then we get, some probes that were not previously associated. So let's look across data sets. So here I have a list of data sets. These are all lung adenocarcinoma patients. In one case, they took normal instead of adjacent samples. So these are not from the same patient, where the adjacent samples are from the same patient. But it shouldn't matter. A normal sample is a normal sample. And so we're going to use these as reference signatures against the panels that I just defined. And look at the beautiful pictures. So I had to break it up on 2 slides, but you'll notice positive panel, great enrichment. Negative panel, great enrichment. Positive panel, great enrichment. Negative panel, pretty good enrichment. This one you're starting to see a little bit of spread out, but hey, it's still statistically significant. This over here is the random model. So same thing like you saw before, we're randomly distributing. Here's the trick. Notice how all these numbers are over 2. Like here I've got 3.6, 4, almost 5. You see where I'm going with this. All of these A through F would show up off the graph. That's how far out these panels are compared to the random distribution that was generated normally. So that's pretty good evidence that these panels are associated with lung adenocarcinoma, in addition to the fact that the literature supports most of our findings already. So similar to what we did in SARS, we started with lung adenocarcinoma cells, but we're not staying there. First, I looked at small cell lung cancer. Then we moved to different parts of the body. Glioblastoma is part of the brain cancer. breast cancer, cervical cancer, prostate cancer, bladder cancer, a whole bunch of GI cancers, adrenal cancers. You get the idea. Comparing those panels across all those signatures, again, look at how far away from the random distribution most of these red and blue dots are. That just gives more support to the idea that these are not randomly generated numbers. We're getting a true signal. And it also suggests that the genes in those panels may be global cancer associations, not just lung cancer. So then we wanted to know which leading edge probes were coming up the most across all those signatures, because those are the probes or methylation sites we want to focus the most on. And there are 10 positive leading edge probes and 6 negative leading edge probes that are found across almost all of those signatures. So here they are. And again, like we saw with the other T-score maps, you'll notice sometimes the statistical significance for a T-score is not there, but it was borderline, so our program was able to pick it up. And I've highlighted 2 genes in here that are of interest. The black arrows point to this PRDM14, which is the top scorer for the positive panel. And this other arrow is this DEFB118. It's the top score for the negative panel. So when we look at those, okay, we find that all of the leading edge genes have an association with at least one type of cancer. So if I look at this PRDM14, it's already reported with pancreatic cancer, leukemia, breast cancer. It's been shown that if you have overexpression of PRDM14, you lose sensitivity to chemotherapeutic agents. I mean, there's a lot of literature on that PRDM14. And it's across multiple cancer types, so it's a global cancer mechanism. Interestingly, a few of our probes are connected with one cancer, but not necessarily the one that we were looking at, or the one that it was found in the comparison. So like if I take the DEFB18, the top score in the negative panel, that's been shown to be a driver in gastric cancer. Cool, we agree with that. But it was never shown in lung cancer. So now we can rethink current therapies. Maybe there's a therapy out there already looking at gastric cancer that's targeting this DEFB118. We could repurpose that now for lung cancer based on what the computer is predicting. So there is a lot of information here in terms of, yeah, all of these genes, all of these methylation sites identified are already associated with cancer. But which type of cancer is new? Or the fact it's a global mechanism may be new. And those are the things that this information can help move forward in a clinical setting. So with that said, I'll take cancer questions with the general questions in just a minute. But I just want to review real quick what we've discussed today. So my overall goal was to elucidate molecular changes associated with disease mechanisms through computational analysis of next-generation sequencing data. And this is to identify potential targets for therapeutic development to overcome disease. We've seen today how you can use gene expression meta-analysis to examine messenger RNA expression of SARS infection in lungs. We've seen how you can use pathway activity meta-analysis on the same type of data to look at antibiotic resistance in bacteria. We've seen how this approach can be used for methylation site identification across various types of cancers. And so with that, I need to thank a ton of people including many that are probably not listed on this slide. But Amber Park and Marius were the two co-authors on my SARS papers. I need to thank my mentors and my committee from Rutgers for the dissertation reviews. And then, of course, Tan Mei and Rid Hammond, which are my co-authors on the upcoming cancer paper that you'll be reading about soon. Okay. And so now I'm done, except for questions. So fire away.
Oh, thank you so much, Professor. This is actually a lot to take in, and quite inspiring. And I'm sure we have a couple of questions from people. But however, I would like to kind of open up the questions by asking you a question or a couple of questions, more kind of generic. So I noticed there's this uniqueness in the approach which you currently using, the computational approach you're currently using. You've used it for microarray data and even methylation data. So I'm kind of curious, can you use this same approach for chip-seq data?
Chip data is a little bit different in how it comes through GEO. So no, this is not horribly adaptable to that, but you can take chip data and possibly convert it into something that is usable. The trick with chip data is you're usually getting a picture type of analysis, so that's going to take a different approach. Anything you can boil down to a signature is something that you can then use in this type of analysis.
Okay. Thank you for that insight.
Yeah.
Secondly, I want to probably on behalf of novices like us, so is there maybe like a tip or a kind of a hint you could give to aspiring bioinformaticians as regards how to go into computational biology and then try to answer meaningful questions? Because cancer is out of maybe like according to the GCO fact sheet, out of 10 non-communicable disease as death that is caused by non-communicable disease, 3 is actually caused out of cancer. And so it's kind of becoming a menace which needs more hands. So do you have a kind of advice or kind of a hint in terms of people that want to dive into cancer or epigenetics in general?
It's such a broad field that I would say, wow. The biggest challenge I find with types of cancer is sample size. For example, I recently published a paper on diffuse intrinsic pontine glioma. That paper was a real challenge simply because the sample sizes were not there. You could get plenty of brain samples from infected people, but you couldn't get them from the normal people. And that was a huge challenge. So fortunately, Gene Expression Omnibus has recently put in a search feature where you can sort data sets by number of samples in the data set. I find using that to be helpful these days. But yeah, I mean, that sample size is going to be your biggest limitation for some of those.
Yeah, thank you, Professor. I think I would like to pause to check if there's anyone. Is there anyone having any question?
While people are thinking of questions or unmuting themselves, I want to point to the GitHub real quick because the code that I use for running this is on that GitHub. So if you're not a programmer, you can go there, download that, and I can kind of walk you through how to use it as you need it. But yeah, just want to point that out to people who were asking me earlier, how do I do this?
Okay. We have a question from Michelle.
We have questions actually.
Questions here that can you do methylation meta-analysis specific to only include known oncogenes of proto-oncogenes in order to compare more easily the known genes associated with cancers?
Yes, you can. The challenge is going to be identifying them beforehand.
Okay. So,
yeah,
also
so
another...
like, oh, go ahead.
Okay, no, Professor, you have the floor.
Oh, I was just going to say something about if you're going to do something like more of a deep neural network kind of approach, you're going to need to do that in terms of whittling down what you're doing. That's, how do I put this? Not necessary as far as this project goes. But anyway, go ahead. Next question.
Okay. Can you please explain how you generated LAM panels twice by inversing the orientation?
Sure. Let me go back to the picture. So we're dealing with two identification signatures. Basically, we took this one data set and we divided it in half. And that was because I had the proper number of samples to be able to do that. So I took one half, made a signature, took the second half, made a second signature. Then after I have my two signatures, I define queried methylation sites for the positive end and the negative end of one of the two signatures. And then I run GSEA. And so that'll give me positive leading edge, negative leading edge. So that I'm going to take the same two signatures, and now I'm going to use the one that I used for the probes, the query sets, oops, go back, for the reference signature. And then I'm going to take the one that I used for the reference signature first, and I'm going to define query sets. And then I'm going to repeat the analysis to get a second leading edge, positive and negative. I'm then going to compare the membership of those two in order to find the genes or probes that are similar, and that's how I'm defining these panels. So before, we only did half of it, and then used the positive and negative leading edge to define the panels. But here, we're doing it twice to try to make it more robust. I hope that helped whoever was asking.
Yeah, I hope so too. Thank you, Professor, for taking our time to answer
I have a question, sorry.
So yeah. I think there are still more questions, but someone would like to ask if it's possible to have a practical session.
Yeah, I mean, it's possible. The $5 question is when I'm fitting that in a schedule. So we can talk about that offline. There is something to consider. I will be putting videos onto the GitHub in the next couple of weeks. And so if people watch the GitHub, the courses repository in the GitHub, there will be videos that will kind of walk you through how to do some of this. And that may be helpful as well.
Okay. Just in addition, I just bookmarked our GitHub page, so I think you can also do all that. So we can all bookmark it, so we could check back every week, every day, every if there are videos uploaded. So
I think if you subscribe, it should email you when there's an update.
An update, yeah. So I think Austin, you have a question?
Yes, please. I do have. I do have a couple actually. So my first question is for the staphylococcus, the resistance presentation. Okay. Yeah. So you identified lysine biogenesis as a major or a key player in drug resistance, and I think it's kind of super interesting to note because lysine is a major target for multiple post-translational modifications of protein. Now, which means that different post-translational modifications occurring on that protein could determine susceptibility and resistance at the same time. For example, if you're looking at lysine molecules for something like simulation or ubiquitination occurring at the same time, or even other types of PTMs could have different outcome when it comes to drug resistance. So I wasn't surprised. Then the approach you described for the cancer, I think we did that. And that was super cool because it was one of the projects that we did, which was using similar computational means to predict not just genes this time around, but we're looking at proteins that are involved in a particular disease, but then we found a relationship between that disease and some other diseases. And I was surprised. I was surprised to see that the genes that code for those proteins were also identified in your analysis. And in this case, we're looking at colorectal cancer, but obviously you're looking at a different type of cancer. And there we proposed these kind of genes, but proteins per se, to be hop proteins, because the interface between different other types of cancer. And yeah, you could see 7 signatures here for colorectal cancer. And when you showed the genes that were overlapping in this kind of, I think the other one, there was a slide you showed. Yeah, this type, yes. So you could easily identify genes that co-occurred in all of these, and this is super cool. I mean, it gives a sense of validation to computational means of performing this kind of analysis. So yeah.
I love it when people tell me I'm right. So thank you for giving me that. This is my mission as a computational prediction person. I'm right. Yes.
Two things that I think you did so well was the sampling and getting the distribution, which means that you're trying to, and again, the number of random samples that you're taking is 1,000, which is significant. One of the challenges I find with sampling is that some people sample once and they get the distribution and they're like, oh yeah, that's fine. And then they move on. Or they sample conveniently. I mean, by using conveniently is that they sample the number of times that they think they will get a certain distribution. And once they hit that, then they try to justify that type of sampling. But yeah, I think 1,000 times is practically unbiased. And if you're going to see some signatures, you would identify them. So that was really cool. And then also plotting the distribution, it's another layer of validation to say, hey, my method worked. So that was good to know.
Yeah. I personally like 1,000 simply because I can do the null distribution p-value. And I find that very powerful. To say something like, I beat all but one out of the 1,000. It gives it that power. Anything less, like if you took 100, for example, then your p-value would be less than 0.01. Yeah, it's significant, but I'll wait 10 minutes and have the computer give me 900 more.
Yeah, that is one. Another good thing too was to adjust for the p-values, which I believe you use the word normalized. So it's more like correcting for this multiple kind of testing in terms of sampling. So yeah, that was super cool. And
good. I'm glad it was helpful. You can make random models out of almost anything.
True.
For the sake of those who are beginners in the area of bioinformatics, so the discussion might seem too advanced. So we've been referred to the GitHub page of Professor, and we can check for more videos as to how to perform this kind of analysis. So, but if you are still new, very fresh, you are just starting out in the area of bioinformatics, don't worry. You can have this knowledge kept at the back of your mind, so that when you get to that stage of knowing all this, the application, but one thing was very prominent after the presentation. Professor was able to tell us that this is the reason why. For example, in the case of a staphylococcus, so we were able to know that, oh, there is a drug that is able to cure or fight against staph despite the the high rates of resistance. So just for you to know the application. So for those of us that have been messaging, so don't worry, we got you covered.
And keep in mind, if you have any questions, I'm easy to find on LinkedIn. You can always send me a private message. My email is there. And if you get to the point where you start using it and you start running into problems, just shoot me an email or a text and I can give you some pointers in terms of, well, your sample size is too low, or you might want to try a different topic because of this biology, etc. We were looking at a preeclampsia the other day using this approach, and we were having some problems with some of the consistency across data sets. And so we started asking the question, well, we're looking at gene expression in placenta tissue. Maybe we're better off looking at some other part of the body then, like if you could get gene expression from the umbilical cord or, blood or what have you, to try to tease it out. So I'm happy to help where I can.
So once again, thank you so much, Professor.
We missed
I don't know,
questions.
do we have any more questions, please?
Yeah. It's from Michelle Hernandez. So she asked that, can you do methylation meta-analysis specific to only include known oncogenes or proto-oncogenes in order to compare more easily the known genes associated with cancer?
Yeah, I think we answered that one already. The answer was yes, but it's not necessary. Okay. It's not going to improve your signal. The only time you're going to want to do that is if you're going for like a deep neural network.
I think that's all.
Yeah, okay. Austin, are you still raising your hand up? Do you have any more questions?
I'm good, thanks.
Okay, I think that's all.
Awesome. Well, thank you so much. It's been an honor to be here.
Thank you so much, Professor. It's actually an eye-opener, and kind of I'm actually intrigued with the fact that there's a kind of a powerful computational modeling that's actually leveraging on the use of T-score and then some other little maneuvering to predict kind of genes, especially in the last category of your presentation on cancer. So kind of looking forward to your videos to see how I can, or we can, I'm sure we can make use of one of those codes and then see how we can actually make some impact in the bioinformatics world. It's actually very interesting, and we really appreciate your time and then the explicit explanations and then answering our questions. Thank you very much, Professor. We really appreciate.
Thank you.
Yeah, so I would like to call on our CTO, our Chief Technical Officer, in the person of Dr. Austin Amakiri, a senior bioinformatician at Procogia, to actually give us a more of juxtaposing, actually. So, Dr. Austin, over to you, please.
Right, thank you so much. Thank you, Professor. And thank you, everybody, for joining. So, I think basically...
Yeah.
Right. Okay, so yeah, I'm smiling because, I mean, Professor touched on almost all the things that I was going to highlight. So to start with, I want to start talking about the opportunities that we can, gain by using very simple, obvious tools within our disposal to make huge predictions or huge impact in the scientific world. So I know that during the course of the presentation or during the actual citation of Professor Comfort mentioned that she leverages SQL, Python, etc. But those are not the only ones. that are out there. For every bioinformatician, they have different preferences. So for me, my preference is to write in R. So all my pipelines, all my code base are in R. That is not to say that I don't use Python. I only use Python when I have to use Python, or when that is the only option I have available. Because yeah, for heavy computational workflows, you may want to use things that you know will run very fast. Sometimes Python might be more efficient. Other times, you could just write simple R codes and call it in Bash. By using Bash, I mean call it from the command line. It will still perform the same analysis. So having said that, I want to talk a bit about opportunities in bioinformatics, because bioinformatics is actually a new theme in science and in research. Everybody wants to get involved in bioinformatics. But the question is, what are the opportunities out there? How do I fit in? Bear in mind, bioinformatics is very broad. No one area fits all. So that is to say that you can carve a niche for yourself in bioinformatics. For example, there are people whose area of expertise is plant bioinformatics. They look at plants using computational tools. There are those who focus on animals. So not everybody has to go into cancer. There are those who work in the industry. So to me, generally speaking, I would define 2 career paths, or 3 actually. So the first is in academia, for those who really love research and really like to work in a research environment. So there are lots of opportunities within research groups for bioinformaticians. That is true, maybe PhD, master's, postdoc, assistant professorship, tenure track professorship, etc. For those of us who don't really like academia too much, but we respect those in academia, but we prefer more immediate impact in the industry. So there are a ton of industries that are recruiting bioinformaticians. Now, she mentioned something about big pharmaceutical companies, but that's not all. There are opportunities in biotechnology firms, government agencies, there are even startups. And interestingly, many data analytic companies are actually recruiting lots of bioinformaticians now. Then the final career path is the fun of entrepreneurship, consultancy. So as the world is moving so fast, research is generating tons of data. And this data needs to be analyzed because we want to draw actionable insight from this data. So some companies actually do contracts, companies or consultancy firms to actually analyze this data. I want to give a typical example. So drug modeling. A pharmaceutical company develops a candidate drug and they want to predict how this drug is going to perform in clinical trials. Now, budget for clinical trial runs into millions and hundreds of millions of dollars. But you can pay a consultancy firm a fraction, 1/10 of a million, maybe 100,000, and in a few months, they will model how your drug or how your candidate drug will perform in the clinical trial and give you a verdict of, hey, don't proceed, it's not gonna work, or give it a try, it's actually gonna work. So that way you save yourself having to go to phase 1, phase 2, and then get to phase 3 clinical trial and then find out that no, the drug is not effective enough, and therefore the money spent is gone. Again, there are also instances where government contracts bioinformaticians or consultancy firms to model disease outbreak. In Nigeria, for example, there is always this incident outbreak of Lassa fever, cholera, monkeypox, etc. But then the CDC pharmacovigilance and I think vigilance team can actually model to tell when the next outbreak is going to happen and take steps to prevent that outbreak. And this type of consultancy played out a lot during COVID-19, where so many different groups are actually modeling to tell how many people are expected to die from COVID-19 if the trend continues, and also to predict how many people are expected to recover based on different modules. And then lastly, you can use this type of modeling to identify candidate drugs. So instead of spending hours and years in the research trying to identify candidate drugs and then only to find out that they don't work, or you spend millions, you can actually model this type of drugs, for example, small molecule inhibitors, screen them, identify how they bind using molecular dynamics and stuff like that, and narrow down the search for drugs to a few hundred instead of thousands that you want to screen in the lab. So you also save a lot of time. So these are some of the areas where bioinformaticians fit in. But I have to highlight a very salient point here. You cannot be a good bioinformatician if you do not understand basic statistics and biology. Now, that something is statistically significant for those of us who like numbers does not mean it tells anything biological. You have to be able to relate it. If you listen to the seminar, the professor tried to relate all the statistical significance that she observed to biology. So that makes you stand out as a bioinformatician and not just a data scientist. You also have to understand the biology of the subject area you're diving into. So you cannot just wake up and start looking at cancer without understanding at least the basics of cancer or the basic molecular undertone for cancer, because that is how you are able to make that establish those kind of relationships. Now, for those of us who are very new to this subject area, that might seem overwhelmed by all of the jargons that we've heard today, don't worry. This is just one step forward, okay? What I would suggest we can do is, one, to pick up a subject area in bioinformatics that you're interested in. If you're interested in cancer, fine, start reading. Read widely, attend seminars like this. No knowledge is actually a waste. It will come in handy at a point when you least expect it. Focus on that area. Try to build capacity. NOBLEKINMAT is providing an opportunity to train bioinformaticians, and that's taking us from 0 to hero. Get involved. If not with NOBLEKINMAT, look for other communities that are willing to train you to answer your important questions. Don't jump in from the middle. You will get lost. Don't assume you know when you actually don't. Ask questions. No question is funny. No question is actually stupid, because to be honest with you, most breakthroughs in research come from asking very weird questions. So ask those questions. They are mind-blowing. They help you think out of the box. Two researchers can perform the same experiment and use the same experiment to answer different questions, and the answers are valid. So don't think, oh, I don't want to ask this question because it sounds this. No, we have WhatsApp groups, we have Telegram groups. You can ask questions. Somebody in that group will know something or would have come across a resource or resources that may help you. We're going to continue to update our website. We're going to continue to put our resources so that they are available to you at any time you need them. We'll provide videos, we'll provide walkthrough, tutorials, and from time to time we organize hands-on sessions or hands-on sections where we show you practically how things are done. But until then, this is the opportunity you have to start asking yourself questions, start asking questions. For me, throughout this seminar, I have a note where I wrote down as many questions as possible. And these questions are not necessarily for me to ask, but for me to answer with research. So it's an opportunity for us to start thinking of what we can use this knowledge to how we can use this knowledge to benefit our immediate community, to benefit our country, our society at large. And with that, I am happy to answer questions. Just don't make it too hard for me. Thank you. You're mute if you're saying.
Yeah. So thank you so much, Dr. Austin, for that insightful journey into bioinformatics. So it actually came in handy for those of us that are already in bioinformatics. So you still get to learn more in terms of career paths as a bioinformatician. And for those of us that are novice, this is also kind of a tap on your back that you can actually get this done. So before we take your questions, so just in addition, you don't necessarily have to have a biology background. If you're a computer scientist or a statistician or whatever, you can dive into bioinformatics. If you have computer skills, then maybe you're good in programming and you've never done science before, you can dive into bioinformatics and you can actually excel easily. And if you have zero bioinformatics skills, you have zero programming skills, but maybe you find yourself in the field of science, you can also actually dive into bioinformatics. When I started, for me personally, like I said, I picked interest in bioinformatics not because I was actually studying bioinformatics in details. It's just out of one of our research paper. So after all the wet experiment, and then we needed to do some kind of analysis. So it pains me that it has to be contracted outside to someone else to do the analysis. And since then, I check the GSE data that that person actually used to analyze the data. I downloaded it, learned one or two things, and then tried to play around with it. And the rest is history. Half of my thesis is actually bioinformatics. So I started learning on my own, just as our CTO has actually advised. And from there, you can also, I would like to add that it's important you seek for mentors. It's important you have a mentor. You might learn passively from a mentor or actively. It all depends on you. So we'll take a few questions, and then we have one or two announcements from our CEO. I remain my humble self, Hamza Mohammed, so the CTO of the COO actually, sorry, of NOBLEKINMAT. So please, do you have any question for our CTO?
And he has begged that you should take it easy on him. I want to appreciate some of the participants here. I might not be able to remember or name everyone, but one of the prominent people I will mention is Emmanuel Amos. So he and his team have been stable with us right from the beginning. And also, I'm not sure she's still around, Marcelina Hernandez. So I'm not sure she's still around. So she has been here with us, and a lot of other people too. I'm sorry if I was not able to mention your name because of the video. So the guys that have their videos on, so I was able to pick them and notice. So thank you so much for attending. So I think
there are two people raising their hands up.
Yeah, Michelle Hernandez. Yeah. Sorry. Thank you for being here. So it's good to have you here with us. So if you have any
two people raising their hands.
Muazzam Muhyiddin, you can, and then Hakram Nadir,
So,
Yes,
Michelle,
yes.
I like your glasses, actually. Thank you very much. So, Muaz Muideen, over to
No,
you,
I
please.
think
You have
Akram
Yes.
wants to talk also.
okay, I think let's start with Muaz first, please.
Okay, okay.
Okay, Muaz, you can.
Okay. Good day, everyone. I just want to seek for recommendations or ask a question. Basically, for someone with a background in biology, I found out that for you to be in this field, you have to learn some programming and some computer skills. So, what will you recommend, like for somebody with a good background in biology and he want to go into this field of bioinformatics?
Right. So that's a good question. You don't actually need programming skills to be a bioinformatician or to excel in bioinformatics. And I will explain to you why. So there are so many tools out there that are web-based. So you basically upload your data, select parameters, and hit run. The only downtime to that is this. Sometimes you only have allocated memory or RAM size, which might not be sufficient for the type of analysis that you want to do. So because of that, people end up or tend to try to write their own programs and optimize it the best they can to run on their machine or on their local clusters. And with the advent of cloud computing now, a lot of all these web servers are being hosted on the cloud. Basically, what it means is that you might just need to pay a subscription fee to use it. Now, if you want to consider programming, that is absolutely fine. The two most commonly used languages in bioinformatics is R and Python, and the both of them are interpreted languages. So basically, in Python particularly, you're writing it more as if you're writing a sentence. So if you understand the syntax, you're almost good to go. And there are so many resources available to guide you. A Google search of any of the questions in Python or in R, you will have so many answers, so many suggestions that will guide you. One of the places I recommend people to start their programming journey is on Coursera. There is Introduction to Programming. I took that course 5 years ago, and it's still relevant to today. So from there, you can transition to maybe more advanced programming skills through Udemy or even in Coursera. And again, there is a universal university on YouTube. There are so many people there that are teaching you a full master's course for free on YouTube. Make use of it. Get your hands dirty. By getting your hands dirty, I mean get yourself a laptop, download the program, and code along. There is also Code Academy. There are so many of them. So you can actually get started. And we're here to support you. If you have a programming question, you don't know how to navigate it, you can also leave a question in any of our communication channels, and one of us will get to you.
So
Okay,
thank you
so
much.
much.
I want to believe the question asked by Muaz has been answered effectively. So we have Balogun Sadiq Murphy. Yeah. You have the floor, Mr. Balogun Sadiq Murphy. Can I ask your question, please?
It's actually a mistake, so sorry.
Okay, it's a mistake.
Yeah.
Okay, you can also make that a question by adding a question mark to it. It's actually a mistake. So that has been answered. It's actually a mistake. So please, any other person, please? Question? Okay, Akile Enoch Olanrewaju. I hope it's not a mistake too.
Thank you very much, sir.
Okay.
Yes, sir. Thank you very much, sir, for. But I have a question to ask. After a biologist, let's say I have a good background in biology now, or let me say, okay. learned maybe Python and maybe, let us say maybe R2 as a programming language, then how can I really further? Because I don't know whether or to go to proteomics, and even if we are going to start on the two, like from that programming language, is it to read some biology textbooks or some bioinformatics textbook, or only little clarity on that, sir?
Yeah, I think that's another good one. Yeah, bioinformatics is very, very broad. People have different interests. Professor Harry, we heard from, her interest is more on genomics. I have a very different interest. I love proteomics more. And most of my research and most of my work is focused on proteomics. That is not to say that I don't do genomics. I do a lot of genomics as well. Now, I don't know it all. I learn every day. So what I did was to pick an area. So I chose proteomics and I started learning everything I can find on proteomics. Now, one good way to learn is through internships. If you have that opportunity, if you have opportunities to volunteer for capstone projects where a particular task is assigned to individual participants, then it's also another very, very decent opportunity to learn. Through mentorships, again, if you have or identify a mentor who is a bioinformatician, you can key in through their mentorship. So you can say, hey, I'm interested in this area, and the person can say, okay, cool. Since you already have a background in biology, can you make this kind of research? What do you find? So that way, you find out that you start thinking outside of the box of a biochemist and more of trying to think universal, because bioinformaticians cut across all of this, bioinformatics and computational biology cuts across all of these life sciences, including computer sciences and more.
Yeah, thank you so much. I want to believe the question has been answered, right?
Yes, it has been answered, sir. Thank you very much. I really appreciate it.
All right. I think I'm going to take the last question from Oluwabumi Ojo. I think I hope I got that pronunciation right.
Yes, he has got it right. Thank you very much.
Oh. You didn't tell me thank you for getting it right.
The session has really been very awesome. I really learned a lot. I would have loved to drop my question in the chat box, but I don't know why I'm not been able to drop any message. It's just saying I'm not a participant. I don't. Okay, so I'm a medical doctor. Currently having my master's in biology here in the US, and I'm working on peptide phage display on pancreatic cancer, and I really love it. I'm currently taking a statistics course where I'm being taught how, and I'm purely a this. So hearing Dr. Austin mentioning a programming that sounds like what I've heard before, I was kind of happy, and I really want to dive into this point. I appreciate that suggestion of going on to Coursera. I've taken some courses in Coursera, but probably I've not really thought of bioinformatics before. So I would want to dive into that. I would appreciate if you can help drop the link to the WhatsApp page so that I can follow up with if I have any question, whatever, to drop there. And I really think that I will be encouraged to follow this pathway. It's really, really, really new to me, to be honest. Even my master's program is new to me, but I think I'm finding it challenging and I'm loving it. Thank you very much.
All right. Thank you so much. It's Oluwabumi. Sorry, Dr. Oluwabumi Ojo. So.
Ojo.
Yeah, she said I got it right the other time, so I decided to get it wrong now.
You now had an
extra
sauce.
Oluwabumi Ojo. You got it right both times.
Okay. See? So, Dr. Austin, do you have any comment for that?
Yeah. So, like I did mention in my initial presentation, I started off by emphasizing the importance of statistics. Because if you don't know what the right statistical test is, then you're very far away from getting anything substantial. different projects require slightly different types of statistics. If you were part of the presentation from the beginning, you could hear me ask a question: why did she prefer the t-score instead of some other more traditional linear regression kind of methods? And again, these methods have all been proven to provide fairly consistent results across board. Now, everybody has a slight different preference. Her justification is that she thinks or she believes that the t-scores work better for large-scale data. So it's something to consider. So it's the kind of argument you want to be making when you're presenting your findings, because as a reviewer, that's the first thing I'm going to criticize, especially if I don't like the method. And trust me, when somebody criticizes your method, your statistics, they are as well as telling you throw away your research. It's useless to me. But when you have sufficient justification for the statistical analysis that you chose, then the person can no longer throw away your research or underrate your research. So that's super cool. Coursera is a good place to start. YouTube University is a better place even, because you can play the YouTube and you can cast it on your TV and just watch. I find it more practicable to learn by doing. So I can play on my TV and then just sit in front of my laptop and try to replicate the things that I've seen the instructor do. So it's one cool place to learn. Another good place to learn is through community kind of learning, where you identify yourself with a group of people that are passionate about the same thing that you are, and you share ideas. You can even create capstone projects, like, hey, can we in 1 week, create this kind of data or analyze this kind of data? It's another way to learn. Again, it's learning by doing. But all of these ways we've suggested are self-paced. So you learn at your own rates. We're here to support you. Obviously, we're here to support you in any way that we can to learn programming. I offer programming classes for free. I also offer kind of tailored programming classes for a fee, but that is not the point. The point is, when you have a question that you think you cannot answer and have a platform where you can ask those questions, then make use of it. I'm sure Ashraf and Hamza will be sharing the links to the WhatsApp groups or Telegram groups, as the case may be. Always review contents from others. For me, yes, I'm a senior bioinformatician, but I also go back to watch videos from other bioinformaticians and I pick up 1 or 2 things from them. And the funniest thing is that you never know when you will need it. So the curiosity to learn from others is also a plus. And I think you're already in the right step. You're taking the right steps and you're doing everything right. So time will tell.
There's another question.
Okay.
Hamza, were you able to see that? Comparison. Sorry, I'm trying to post some updates.
Nope. So, which question?
It's in the chat.
By who? Still checking.
Okay, the question says that, can you explain the difference between sequencing and comparison using t-score in bioinformatics? The differences.
Sequencing and comparison. Right. So sequencing, it's basically a platform, sequencing platform that you use to extract information about a sample. And the type of information you extract are basically the transcripts, which is the RNA, the transcripts contained within the organism, which is like the RNAs. It could be messenger RNA, it could be any other type of RNA, microsatellite RNAs, etc. Or even the ChIP-seq. However, t-score is a statistical, so let me put it this way. So first one is a platform. You use it to extract data, while the other one is a statistical parameter that you use to draw inferences based on the data you extracted using this sequencing platform.
I think that's it.
Thank you very much, Dr. Austin. So I think that's all for the questions. So before we go, I would like to thank each and every one of us, and then I would like to call on the CEO to give us some announcements and then wrap it up. CEO, over to you, sir.
Right, thank you so much, everyone. It's really been an honor, and we appreciate everyone for being here. I'm so much happy and elated about the way the participants have been active, and those that were not able to even ask questions are staying and being online, and they are stable and asking questions and following the trend. So I've been watching the trend also on our social media, other social media platform, on Facebook Live. So, and it's a promising community. So just like Hamza said right from the beginning of the seminar, we are creating a movement because we saw the need for this kind of skills to be imbibed in our culture, because the COVID-19 pandemic has taught us a lot of lessons about how this biological information, bioinformatics analysis is so important to our survival as human beings. So we know how important it is to medicine. We know how important it is to even agriculture, life sciences. So, but it's so sad that a lot of people do not know even the most basic level of a bioinformatics analysis. So that is why we are taking up the challenge. So, and we want everyone to join us in that challenge. So yes, we are just starting out like in a big way. We would have some lapses, we would have some places we need to improve on. So that is why we are sharing. We shared a form for you to fill. It's in the chat box, so you can check and you can fill in the form to give us more feedback about what you think we can improve upon. So that is that. And the second one is that we also have our social media presence. So we are heavily present on LinkedIn, we are present on Twitter, we are present on Facebook. So, and our WhatsApp groups are also vibrant. Right now we have 3 WhatsApp groups. So that is why we are now migrating gradually to Telegram. But we discovered a lot of people find it easier to use WhatsApp than Telegram. So that's why we are still maintaining the WhatsApp group present for now. But we have the Telegram page. So, and the Telegram pages will be used to share different materials like the textbooks, the materials, the video resources, the audio resources, or any other materials. So as regards bioinformatics. So now that is that for today. Then next week, same time, the time and the schedule will also be shared to us. We plan to have our website ready by next week. So we plan to have our materials uploaded by then. But if we are not able to have the materials uploaded by then, so probably we'll organize another live session whereby we'll be treating all the hands-on practicals. So the genomics, the transcriptomics and epigenetics, and also the metagenomics. So we have facilitators that are in charge of that. And that's going to start by next week. So we want every one of us to participate actively in that sessions. So more than 1,800 people registered for our classes, but it's unfortunate that not everyone is here with us. So we want everybody to be part and parcel of this movement. So we are using this medium to inform us that we should also implore our friends, our colleagues, and those who we know that these skills will be beneficial for. So likewise, so we plan to go in depth into because we are building a movement. So we need partners in different universities, in different institutions, whereby we can bring this gospel of prosperity, of impact to them. So because we know that how do we spread this information is just to take it to the source. So we know that you are the link. You are our link to the source. So we are willing to come and spread that gospel of bioinformatics to wherever you are. And so we are seeking for partners in educational institutions, research institutions. So we are willing to come and partner with you. So if you know that the messages we have, the skills that we are willing to share, so if you know that it will be useful for a lot of people that you are in the midst of, or you belong to an organization you belong to, please feel free to contact us. The email to contact us or our social media platform is widely available. So I'll drop the email now, noblekinmat@gmail.com. So you can send us an email there. So shooting us your request so we can organize and meet with you. So we sincerely wish to make it known, because our goal is to have more than 1 million bioinformaticians by the end of 2022. We know it's a very Herculean task, so we need you to help us fulfill that goal so that we can create a society of impact, prosperity, and sustainability. Thank you so much for being with us all this while. We really appreciate your presence, and I say I appreciate you once again. So over to you, the CEO. Hamza.
Thank you very much, CEO, for the announcement, and thank you all for joining us. We hope to see you again sometimes next week. Have a lovely day.
Now, the meeting is officially over.
Thank you. Bye. Bye-bye.
Bye-bye. So the recording of the meeting will be sent to our emails. So watch out for that too.
Thank you so much, sir.
You're welcome.
All right. Thank you so much, sir.
Bye-bye.
Bye. Thank you. Yeah.